I will today focus on one of the activities and infrastructures that we are building in the Netherlands to improve cancer care. And this is a setup that is organized within this Hartwig Medical Foundation. These are my disclaimers. And I would like to start with my interpretation of what the promise is of personalized cancer treatment, or what personalized cancer treatment would be. For me, that would mean that if we can give the right drug to the right patient, the right dose, from the right moment onwards till the right moment. So that sounds good, I think. And of course, we are aware that we are not there on many different aspects. Basically, what current clinical care quite often is, it's a one-size-fits-all type of treatment. And we want to move away from saying a cancer patient with a certain disease gets first therapy A, then therapy B, then therapy C, towards a more informed decision making where we use biomarkers to stratify patient towards the right therapy. Of course, that does not answer all these challenges that are over there, but this is the one that I will be focusing on today. So where are we today? Again, focus on DNA-based personalized treatment. This is an unreadable table for you guys, so that's good news, because that means the table is long and it is growing. So the number of biomarkers that we have for certain drugs, or the number of drugs that we have based on specific molecular pathways, is growing. So that's the good news. I would argue the list is not long enough. I think most of us will agree, looking at mortality rates in cancer patients. The other part of the good news is that we now have the technologies made possible by companies like Illumina to systematically measure DNA using next generation sequencing technologies. This is all background. This has also attracted a lot of attention and sparked a lot of initiatives worldwide. Genomics England will come up in the next presentation, so I'll not say anything about that. And of course, we've all heard of the US Precision Medicine Initiative, bringing along huge promises. Basically, these combinations, the targeted therapies, and the possibility to analyze DNA systematically brings along huge promises. Good. So where are we today when we look from a patient perspective with these promises? Here I give a hypothetical patient report based on this current knowledge, on this previous list, DNA markers. So let's assume you sequenced a patient, a tumor, completely, and project knowledge on top of it. This is what it will typically look like. In this case, a female breast cancer patient, and we find the only thing that is found when we look at actionable mutations, a HER2 amplification in this case. So that's good. So this is already current practice. And in most labs worldwide, or most institutes, these tests are done, not necessarily by next generation sequencing, but by a method. So we don't, well, we may gain a little bit because the number of indications is increasing over time that we have to test for, so next gen sequencing is handy, but there's a lot of techniques that can test for this. However, what we stop thinking quite often here, and we just give this therapy and forget about it. So established therapy is just given without a lot of evaluation. And we should be aware, depending on the treatment, which costs money, causes side effects, only works in approximately 10 to whatever percentage of cases. So there is space for a lot of improvement over there. The second thing that we should realize is that quite often we don't find an approved biomarker-based therapy. Yes, we do find mutations. Yes, they are in cancer genes, but no, they are not actionable. And if you're very critical, and if we look at our data, I think only in 5% of the cases we do find a real actionable mutation. And this is metastatic disease, etc. So we're not analyzing primary tumors. So that's another challenge. So we don't have enough biomarkers, or we don't have enough therapies that we can give. It's a combination of both. However, where does the good news come from? Otherwise this becomes a negative story today. There is, in all cases, for every single patient, there is a long list of mutations found in all kinds of genes. And there is a lot of knowledge around these genes from experimental environments, or sometimes from preclinical and from case studies. And this is in 100% of the cases. So if we do sequence, we always find off-label indications that may be useful for guiding experimental treatment. So that's where a piece of the promise is. These are the challenges that we would like to address in the Netherlands, and for that, basically in 2010, long before the US and UK initiatives became public, we started the Center for Personalised Cancer Treatment. This is a bottom-up initiative, so there was no government agenda that said we're going to do this, we're going to focus on that, that's where the promise is. No, that idea basically came from clinical and preclinical researchers that said, yes, we have to do this. And if we want to do that, we can only realize that by collaboration and not just as a single group start working on these promises. A little bit about the Netherlands. We have 17 million inhabitants and about 100,000 novel cancer patients per year, and there is 87 hospitals in which cancer patients are treated. So that's basically our playground. There is 9 academic medical centers, 21 larger teaching hospitals, and the rest are peripheral general hospitals. We started off with 3 centers doing this because we had to do a lot of things together. Unified procedures, we had to agree on how we do things, we have to figure out stuff, etc. So start small, but not just by yourself. That was sort of the message. That was in 2010. We always had the ambition to scale towards the whole of the Netherlands. In 2014, all academic medical centers in the Netherlands have joined this initiative, the oncology departments. 2015, we've been expanding with the large teaching hospitals, and we're now moving towards all hospitals to basically try to organize the first steps on realizing personalized cancer treatment and improving personalized cancer treatment in the Netherlands. This is an experimental setup, let that be clear. So we're not aiming for doing diagnostics in this setup, because that is done in all these different centers, also in coordinated ways. But we aim at improving care. So the first aim of the study of the Center for Personalised Cancer Treatment is to reduce overtreatment with existing targeted medication. So reducing side effects for the patient and reducing costs. And this is a retrospective study setup. So and I'll come back to the setup in a minute. The second aim is to increase treatment options for patients. So can we identify biomarkers in patients and couple these biomarkers to experimental use of drugs, off-label use of drugs? That is a prospective study setup. We've made some choices because this is a bottom-up initiative, so there is not a precedent that says I put $1 billion or euros in something, so we have to be very pragmatic and start somewhere. We said we're going to start with metastatic disease patients for 2 reasons. This is where the largest unmet need is. There is a lack of treatment options often, and this is also where novel, quite often expensive drugs are entering the scene, and we have a challenge incorporating that financially in our care system. So there is really a need from a financial perspective there as well. The approach that we're taking is to collect pre-treatment biopsies. And it should be clear that for metastatic disease, this is not, in most cases, a routine procedure. Typically, no biopsies are taken in a metastatic situation. It depends a bit on the indication. With lung cancer, this is increasing, but it's not a routine procedure in most cases. So that means this is an extra handling that you'll do on the patient, and that means it has to be done in a clinical trial and needs to be organized. And then we've chosen the main focus is to use whole genome sequencing, so collect as much information as possible and integrate that with clinical data. This is basically what the study setup looks like in practice from a patient perspective. It starts with first is a patient eligible for the study, so the patient should be qualified already for going on a certain targeted or a certain treatment. We've broadened the criteria quite a bit. This is a dynamic list of treatments that are given. Patients should give consent, and then a biopsy is taken, and basically this goes to a central facility, I'll show that in a minute, where whole genome sequencing will be performed. In the meantime, the patient will go on the treatment and follow the treatment. Response assessment will be done after about 2 months of time, so then we know if the patient responded or is progressive, etc. Basically, this is the retrospective part of the study, where basically response assessment and genome sequencing results can be coupled with each other, and we may identify resistance predicting biomarkers, etc. Simultaneously, there will be so this is basically a retrospective part and a cohort type of study, and simultaneously there will be a patient report generated which will be evaluated in a molecular expert board or a molecular tumor board, which may provide, if the patient is progressive, options for enrolling in a second study for a follow-up treatment. And these could be these experimental treatments, experimental drugs off-label use, and for that a new study has recently been started. I'll come back to that in a minute as well. Before going, when a patient is eligible for this study, for a follow-up treatment, there will be another biopsy taken just before starting the following treatment. So basically that becomes a post-treatment biopsy for the first study and a pre-treatment for the following treatment. So that also may provide information on tumor evolution on treatment with these targeted agents. Okay, well, we can have a long debate basically on, and I just put this slide in because this discussion or question comes up quite often. Why doing whole exome or whole genome sequencing? A lot of these things can be done with targeted tests as well. For the current patient, targeted tests excellently well suited for answering questions. However, I would argue that the benefits for the future patient is quite often limited. You only measure those things that you want to know. So you're not measuring the things that might be useful for better predictions. And that's basically why whole exome or whole genome sequencing come in. That for the future patient, the benefit should come from there. Of course, there's a huge cost difference in these two tests. However, putative future savings may come from this part if we can better predict, if we can prevent overtreatment, if we can prevent that we give non-working drugs to patients, we may save money. So yes, this requires an investment here, hopefully to end up there. Of course, that needs to be proven, and you can only prove it by doing it, right? You cannot predict that. Summary, I would say, between these two parts, I don't want to pick a left or right. I think you just have to think on what are you trying to do, which type of test is best suited. Targeted tests, targeted next generation sequencing tests make excellent care, I would argue. The benefit from these approaches is that we can learn something for the future patient and has a very high research value. Okay, how does that work in practice? The studies that we're doing, all the patient part is coordinated through the Center for Personalised Cancer Treatment. There's patient inclusion is coordinated, clinical data collection is done in a uniform way in all these centers, and biopsies are taken according to unified protocols, and there is always a tube of blood taken as well. This part is financed through projects and partially through philanthropy from a specific foundation that is supporting these activities. The samples are going to a central facility which was recently established. This is Hartwig Medical Foundation, and this has been established because of the scaling of the activities that we're doing. So originally this was done basically in my research diagnostic lab associated with my research group, and that is of course not where you want to be if you want to scale this to a national center. So that's basically where an independent foundation has been set up with representation of all stakeholders, Hartwig Medical Foundation. That's where histology and DNA RNA isolation is done, whole genome sequencing is done, and the data is integrated in a database and made accessible again to the participating centers. It should be mentioned that this has only been made possible through philanthropy, through a very large donation from a single person. Otherwise, this would not have happened, unfortunately. It should be clear that philanthropy is ending at some point. So we have to find ways to implement this whole setup in routine care. Of course, that becomes easier with success, but that requires time, and there will always be a fight on how to establish and maintain that on the longer term. What is this Hartwig Medical Foundation? It's a non-profit organization. We opened the labs in October last year. As I mentioned, 13 hospitals are already actively providing biopsies and data to this center. 15 hospitals are in the process of joining, and all the legal work is being wrapped up at the moment. And another 20 are lined up to join as well, which basically gives us more than half of all the hospitals in the Netherlands contributing to these activities. Small company, just 10 employees. The setup, there's only one thing, and that's whole genome sequencing. So that makes it an easy workflow in the lab. It operates an Illumina X10 setup, which has the capacity to sequence 18,000 genome equivalents per year. We're working towards ISO certification in this lab for both the lab and the IT procedures. And so far we've sequenced about 350 patients, and that is tumor normal pairs. And I come back to how we do the sequencing exactly. And we're now including approximately 20 patients per week. So the limiting factor, as you can do, if you can do the math, is not the sequencing capacity, is really getting the biopsies from the patients, the informed consent, all the procedures properly done. Hartwig Medical Foundation aims for a national positioning, and although it originated from the Center for Personalized Cancer Treatment, it does not have an exclusive relationship. So we are setting up collaborations with existing national cancer registries and initiatives as well in different areas, and they're in different stages of progression. So this is another way of making sure that all data will be connected and collected in a uniform way. There's a limited set of criteria for enrolling. Whole genome sequencing, yes, it can be done on FFPE material. Yes, you get sequencing results, and yes, there is a lot of noise in your data as well. We find that if you want to have a high quality database, it's worthwhile investing in the samples. It's not really like rubbish in, rubbish out, but if you can prevent inferior quality, you have to do it. And that's basically what we do. We have to do this in a study setup anyway, so we can drive all the labs towards collecting fresh frozen material. And that's a requirement. We need at least 30% tumor cells because, well, yes, you have to sequence much deeper if tumor percentage goes down, and that comes just and then cost will go up linearly. So that's also a thing that we want to avoid. There should be a germline control available. It can be blood or a piece of skin or whatever, and that is required for the bioinformatics. Otherwise, you don't get there. You get all kinds of individual-specific or population-specific variants added to your somatic variant list. Of course, the samples should, there should be a scientific question or a clinical question towards the sequencing, pretty obvious. It's not just, I have samples, just start sequencing. You need a question with that. There should be sufficient clinical information available that can be added to the database, because a database with just sequencing, tumor sequencing data, has very limited value. It really gets its value from all the clinical data that is added to it. And a very important one is that there should be informed consent, not only to allow whole genome sequencing, but also to allow data sharing, because the objective of this larger database is not to keep everything inside, but to share it to make research possible. And there is really explicit consent required from patients, at least in the Netherlands, for allowing to do that. This all should generate a central database for research. We're collaborating closely together with some with clinical genetic groups as well, and with specific research projects also related to cancer research. For example, the Hub Foundation, which is a research organization that focuses on organoid derivation from tumors and doing in vitro drug screens. So you cannot do just one clinical trial on a patient, but you can basically test all drugs and drug combination in a Petri dish. So that might be that data deriving from that. We aim for integration and coupling to this database as well. So you get in vitro, let's say organoid like me type of questions can be asked then as well. Which organoid did respond with what genetic profile to what drug? And that may eventually be guiding when sufficient evidence is gathered in the future for treating patients as well. Okay, this is what the setup looks like in practice, in the lab process. We have blood. We recently started collecting this centrally and collected in such a way that serum is collected separately. We don't have a research plan ourselves. We consider that as a valuable resource to which you can go back and answer questions, do validation studies, etc., for parties that are associated with this initiative. So this is just stored. The pellet is used for DNA isolation, and the DNA, the germline, will sequence to 30x depth. The biopsy, as I mentioned, fresh frozen, first assessed by histology that it should contain at least 30% tumor cells. And we currently we are just doing DNA isolations, but we have set up all the procedures for doing simultaneous RNA isolations from these tiny core needle biopsies. So that does work. We do get sufficient material to allow for whole genome sequencing and RNA sequencing from the same biopsy. The tumor, we sequence to 90x depth to account for the tumor percentage and potential heterogeneity. Of course, yes, deeper would be better, but as I mentioned, costs go up. RNA sequencing, that is still in the pipeline, but that single end, this would just be a quantitative measurement on gene expression that we want to initially start off with, doing single end sequencing. Then the data is analyzed. We do a germline or single sample calling actually on both samples, and that is also used as input for somatic variant calling. So we both get germline reports and somatic variant reports. This is a data processing pipeline. This is complex, I can tell you, and this is also a costly procedure. If you do procedures like this, comparing a 30x and 90x sample, and you use these type of tools, so this is BWA-based mapping and GATK-based indel and variant calling. This already takes almost a day for a single sample just in getting samples through. Of course, you can scale it and run many samples in parallel. But if you add in the somatic variant callers, we have a challenge because they are still immature at this stage for whole genome sequencing data. And you cannot just call variants in one sample, call samples variants in the other one, and do a subtraction. You go wrong because there are, well, maybe in a control sample a variant does not meet one of the criteria like the coverage. But there is evidence that there is reads showing that there was already a germline variant. It would be called as a somatic one incorrectly, very easily. Not a huge problem if you look at small panels, but if you look at the whole genome, this adds up very significantly, these cases. So you need sophisticated callers that take everything into account. It's early days. Actually, there is 4 somatic variant callers in this list here. And I'll show you which ones which we're using, and there is more being developed. And as I said, this is early days, and it's very important to keep following this because I'll show in the next slides that we're not there yet with these somatic variant callers. Actually, this procedure takes 2 days to run all of these tools. So we're basically up to 3 days of time on a large clusters, and I will not mention the cost, but that's very significant as well in this setup. Of course, copy number variation, very important as well. Depends on the resolution that you want to see, what tool you want to use, how much it will cost. And another part is somatic variants, let's say translocations, etc. There's different tools there as well. Computationally very, very expensive for doing at the whole genome scale. So there is a lot of things being developed at this area as well. We're not doing much ourselves there. We keep following what's happening in the world, what other centers are learning and adapting, and we try to implement the community standard as soon as we feel comfortable with it. Okay, this is a bit on validation. Some numbers in it. We use a genome in a bottle, a control sample, one of the HapMap samples. And this is just to show single nucleotide variants in indel. So this is germline variant calling, use BWA and GTK, and X10 generated data, 30x coverage. This is just to show you on coverage. If you increase coverage to 60x, it doesn't do anything with these numbers, and it doesn't do anything with the genome that you can cover. So you cover basically typically 97, 98% of the genome with this type of sequencing, and you don't increase it by increasing depth. If you increase depth, the number of true positives that you detect only very marginally increases, and you see the false positives actually going up. And there is explanations for why that is, and that is because we didn't tune our parameters to higher depth. We keep using the parameters that were optimized for 30x coverage, right? So higher coverage may increase a little bit, but not too much. So what do we look at? I think these numbers are actually pretty impressive. This is looking for single nucleotide variants and indels. We have a precision of 99.7%. For the SNVs and indels, it's slightly lower, and the sensitivity is also above 99%. So I think this is good. If you look at variants in exome, these numbers go up even further. So this includes basically all the whole genome. Then the somatic variants. So we did a validation experiment on that, and because there is not a good reference available publicly yet, we again used these genome in a bottle samples and just mixed two samples at 30% versus 70%. And of course, we didn't realize completely what we did until we started running some of these tools, because this basically gives you 1 million variants. These are related, sort of related Caucasian individuals. So you expect about 1 million variants, and that's much more than you would typically see in a tumor. And some of these tools really go weird if you feed them with so much variation in a sample. So
smoke out of it.
Yeah. But yeah, unexpected thing. A lot of smoke. Yeah, and amazed also from the bioinformaticians, not just the machines. This is a long table. I just want to show you, if you look at sensitivity from different tools, this is for the SNP, the single nucleotide variants. You see here only a sensitivity of that 15% of the variants are found back with this tool, while another tool gets 93%. However, if you go to the indels, you see the numbers are quite different from the different tools, and you see basically other tools performing very well for indels and very poor for single nucleotide variants. So Strelka appears best for the single nucleotide variants and VARScan for indel results. However, the best results come from combining these tools. So that's why we run 4. We have to run 4 to get to sufficient sensitivity. And actually, sensitivity is almost 98% for the single nucleotide variants and indels if you combine tools. I think that's not too bad from where we stand. Of course, there is a lot of improvement still required because this should be done better. Again, for exomic coding sequences, the numbers are actually better than what is shown here. And that's because a lot of these things go wrong when there is repetitive regions, etc. in the genome. But it's basically what you get. This is just to show you how these tools perform. This is for indels, and that's for somatic variants and indels, and here's the combination. So there is only this part of the known true various somatic variants that we should have detected are detected by all 4 tools. So if we would have taken the criteria and, then it would only be approximately, 13% of the variants that we would have called. Take 3 tools, it's doing pretty well, but for the indels, it's actually pretty poor. There is actually not a single variant, not a single indel that is called by all tools. But that's because one tool doesn't call anything at all in the current settings. That's logical. Good. That is just to show you a bit behind the scenes on what is happening. 5 more minutes? That's good. We'll manage that. Data is stored centrally in an anonymized way. Ownership stays with the patients and the hospitals that are providing the information, so it's not owned by this organization. Supplying centers can always get to their own data, and the database is accessible for research. This foundation itself does not do any research. It makes research possible. However, data cannot be just thrown on the street because of all the informed consents and regulations, so there is an independent data access board that keeps an eye on that. Okay, what does this look like? That's good for the last 5 minutes. Because this is a complex process on bringing data resources from internal processes and external sources together with the goal to generate, on one hand, a patient report, and on the other hand, to build up a database that allows biomarker discovery. And we started off doing a pilot with Illumina with the NextBio. Actually, we started off the pilot with when NextBio was NextBio and not Illumina, to basically address these 2 challenges because they offer a platform that makes both activities in principle possible. So we show you a bit of screenshots from that pilot. Here you see this coded patient, a colorectal cancer patient. You can bring in a lot of patient clinical details. You can then bring in a lot of data on treatment and treatment response. The nice thing is you can also show that on timelines. So you see previous diagnostics, previous treatments. This is the treatments for which the patient enrolled, and the follow-up, the vital status. This patient died at the end. So all of that data can be integrated in this platform and used for mining, and also analyses and correlation analysis. Important part, of course, is to be able to put in the molecular data. You can put filters on it. And in this case, it's basically the actionable mutations that were selected. You see this patient has a KRAS, a PI3 kinase, and AKT1 mutation. then you can push basically the button therapeutic guidance, and it tells you, well, KRAS mutation, cetuximab would be the standard approved therapy that you could give. So that can be done just by clicking. You don't need the bioinformatician, data's pumped in, any clinician, any researcher can click through. And it provides this list based on a knowledge base which is present in this software, an off-label biomarker-based therapy list that can be input for discussion in tumor boards or whatever. To be able to act upon that, we've set up this new study, as I mentioned. It's called the DRUPS study, that we are not only see this list of off-label therapies, but that we also make it possible in the context that we're having to provide these drugs. So we negotiated with pharma companies, and several of these negotiations are still ongoing, but from AstraZeneca and Roche, they have provided drugs and molecular indications that we are allowed now to provide these drugs to other patients with other types of tumors. And that is a study that we're doing also in collaboration with ASCO. It's called TAPUR in the US. They have exactly the same setup. They don't do the same molecular analyses, but it has the same goal. And we want to learn from each other that we're not trying a drug because a molecular marker was there and do it and doesn't work and forget about it. We want to do this very systematically, starting with cohorts of 8 patients, provide per tumor type and molecular profile, provide a drug, see if there is a patient responding. If it does, with certain criteria, we expand it, and then there should be more patients responding, etc. So trying to build up this knowledge is not really a traditional trial setup, but it may result at a stage where we say, yes, now we should do a trial for this drug, that molecular indication, and that tumor type. Yep. Explorative study. It recently opened just a month ago in 12 centers in the Netherlands. Good. Then a little bit on the public part, on the cohort part, same database. You can go basically to select multiple patients. There is already something like 13,000 public data sets or patients in there. So you can select by tumor type, analyze your own data in the context of public information, or you can analyze just the CPCT specific data, or you can select by whatever other parameter is in the system to stratify your patient, and then dynamically start doing the analysis. So you don't first have to set up a cohort and it becomes fixed. You can say, now let's just look at the males, and it recalculates everything in the background. So there's a large compute infrastructure behind that to allow to do that. So if you do a selection, whatever it is, in this case the CPCT patients, you can get an overview on which genes are most frequently mutated in your data set. You can click on a gene and get a plot on where the mutations are, and export lists. Every page has a button to export to your own preferred format, so you can analyze it also in other platforms. So this was a very important parameter for us, so we should not be stuck into the system. And output per tumor type and mutation distribution, co-occurrence profiles, you know all these plots. Co-occurrence enrichment depletion plots are automatically calculated, so in this case you see what you know. There is APC and P53 are co-occurring, but BRAF and P53 are not co-occurring in this data set. So eventually what you want to do with the database is answer the following question. For any given treatment Z, are there any mutations, mutated genes, pathways, or any other features that discriminate responders from non-responders, because that becomes a biomarker. And you can keep playing with your data set, because you can ask this question on the whole data set, on a specific treatment, just on the males, on age, etc., to ask this question. And you, as a clinical researcher, not just a bioinformatician, you can play with this data and answer, let's say, the first line of questions to these data sets. However, it should be clear that this only works with a large enough database, and this needs an investment. So I cannot show you anything of this promise because the database is not large enough. But we have to start somewhere and start building on this database. So this is a long-term effort that we try to get established jointly in the Netherlands, but also by collaborating with other international efforts. With that, I think I've nicely filled the 5 minutes, maybe a little bit more. I want to thank, of course, there is no names on the acknowledgement slide because I need another 30 slides to basically fill in all the names on people contributing to this. So there's a lot of participating hospitals, which is still increasing, the funding agencies, and of course insurers are basically becoming involved in this as well. On the longer term, they need to be heavily involved in this. and of course all patients and families that contribute their valuable material and data to this initiative. Thank you for your attention.